So this experiment involves the use of an agar that has been stained with a blue indicator to investigate diffusion. So, hydrochloric acid can be used to diffuse into a blue stained agar in which it changes its color from blue to yellow. We're going to be investigating the effect of different concentrations of hydrochloric acid on the distance that this acid diffuses into the agar. This is what we call the diffusion distance, okay? So we've been provided with a couple of materials as listed in the table. For starters, we have 30 centimeters cubed of 2 moles per deciliters cubed of HCl. Then we have 10 centimeters cubed of an unknown concentration of hydrochloric acid. So these two both contain HCl, one with a known concentration, one without. And then we have 50 centimeters cubed of distilled water, and then 6 test tubes which contain blue agar. If any solution comes into contact with our skin, we are to wash it off immediately under cold water, and we should have eye protection on at all times, okay? So what we're going to be doing is we're going to be preparing different concentrations of hydrochloric acid. I'm assuming based off of this standard solution, we will be producing dilutions through a dilution series to produce different concentrations. Then it says we're going to measure the diffusion distance of each concentration of hydrochloric acid after 1, 10 minutes, and then 2, 20 minutes. Then we're going to use our results to make an estimate of the concentration of the unknown acid. So once we produce diffusion distances for each of the concentrations, and we find the diffusion distance for our unknown concentration, we can compare the diffusion distances and accordingly find our estimate concentration. So it says here we're going to carry out a serial dilution using the 2 moles per decimeters cubed of hydrochloric acid, and we're going to reduce the concentration by a factor of 10 between each dilution. We need to prepare 4 concentrations in addition to the original one, and after the serial dilution is completed, we need to have 9 centimeters cubed of each concentration available to use, okay? So we have a figure that we need to complete to show our serial dilution. 3 things we need to have labeled is 1, the volume of hydrochloric acid being transferred, 2, the volume of distilled water being added, and 3, the concentrations of the hydrochloric acids being produced, okay? So they did say a reduction by 10, a factor of 10 each time, and they've started us off with 10 centimeters cubed of our standard solution. So we know that each time we make a dilution, that we have to have 10 centimeters cubed in each one. Okay, the first thing to know is that we've used 0 centimeters cubed of distilled water here and 10 centimeters cubed of our standard solution. So that's our solution number 1. That's our H from, oh, it's right here actually, this. Then what we need to do is we need the next solution to be 1/10 of that. So we need to go from 2 moles per decimeters cubed to 0.2 to 0.02 to 0.002 to 0.0002. That's what a reduction by a factor of 10 means. In order to do this, we need to take HCl or hydrochloric acid and make it 1/10 of the next solution. How are we going to do this? What we're going to do is we're going to take 1 centimeters cubed of our 2 moles per decimeter cubed HCl, and we're going to transfer it to this next solution, and then we're going to add 9 centimeters cubed of distilled water. Notice how they add up to make 10 centimeters cubed. Now that we've only transferred 1 centimeters cubed of the original solution, only 1/10 of the solution is HCl. Therefore, we have gone from 2 moles per decimeters cubed to 0.2 moles per decimeters cubed. And then we just keep doing that. Transfer 1 centimeters cubed of our previous solution and make up 9 centimeters cubed of the solution distilled water, and then just keep going, okay? So where do we get the marks for this, right? So this is a 3 marker. Our first mark is for correct concentrations. Our second mark is for transferring 1 centimeter cubed of the previous solution to the next one each time. And our third mark is for adding 9 centimeters cubed of distilled water each time. Okay? Then we are told to carry out the following steps. So we prepare the concentrations the same way we set it up in the diagram. Then we label one test tube in the containing blue agar U. Okay? Then we label the other test tubes with their concentrations. So the 2 moles per decimeter cubed test tube will just have 2 moles per decimeters cubed labeled on it. And then steps 4, 5, 6 are simply us adding our solutions to each. And we're doing so by adding 1 centimeters cubed only. So 1 centimeters cubed of our original H, 1 centimeters cubed of all the other concentrations, 1 centimeters cubed of our unknown hydrochloric acid concentration. Then in the following steps, we're going to be waiting. So it's recommended that we do other parts of our question 1 while we wait. But basically, we're told to start a timer, wait for 10 minutes. Once the 10 minutes are over, we measure the depth of the yellow agar, the diffusion distance, for each concentration and then record our results, okay? So right here is a diagram, right? So we have our blue agar and we have our HCl. Initially, there's only going to be these two present, but with time the HCl is going to diffuse, and as it does that, it turns the blue agar into yellow agar. The agar is not increasing in quantity, it's just changing colors, and that is like sort of it kind of determines the diffusion distance when we measure the distance or the length of the yellow or the depth of the yellow agar. Then what we are told to do is continue timing once the 10 minutes is up for a further 10 minutes, so that makes 20 minutes in total. And then once the 20 minutes are up again, we measure the depth of the yellow agar and record our diffusion distances for each, including the values for U as well. Then we're told to record our results. Okie dokie. So for our results, I have created this table. Now, the only things we recorded in general were the concentrations and the diffusion distances, once after 10 minutes and then again after 20. This is a 6 marker. The first mark is for our independent variable, the thing that's changing, the concentration of HCl plus its unit, moles per decimeters cubed. The second mark is for our dependent variable, our diffusion distance plus its unit, as well as including after 10 minutes and after 20 minutes, okay? Also, to mention, you don't get credit if you include the units within the body of the table. They have to be within the titles. The third mark is for there being a measurement at 10 and at 20 minutes for each. The fourth mark is that the diffusion distance recorded for the highest concentration being 2 moles per decimeters cubed, at 20 minutes has to be longer than for the lowest, which is this one, at 20 minutes. So as you can see, yeah, the diffusion distance is a lot greater. 18 is greater than 4. The reason for this being is because it's only realistic for a higher concentration to diffuse a lot faster because there's more particles per unit volume. And then for our fifth mark, our diffusion distances need to be in whole millimeters. Millimeters because the diffusion distance is very short. Whole because that's as precise as you can get with your ruler. You can't get more accuracy than that, or else you'd be making up data. And finally, our sixth mark, our value for U, should have a diffusion distance recorded that is between our value for 0.2 and 0.02. Which, yes, ours does lie because I had to make it that way, but just bear in mind, you're likely to have a value that lies between 2, and it can't be smaller than the rest or greater than the rest, or that could be the case, but it's unlikely. Okay? If we move on, it tells us to explain how the concentration of hydrochloric acid affects diffusion distance. Well, just by taking a look at our table, as the concentration increases, the diffusion distance increases as well. But we are told to explain, so that's not enough. So really, the higher the concentration, the steeper the concentration gradient is, because obviously particles move from a region of higher concentration to a lower concentration. The more concentrated an area is, the steeper the gradient that it goes down. So the greater the contrast, essentially, and therefore the greater the diffusion distance. It then tells us to suggest one error in the procedure described in steps 8 and 10. If we take a look at steps 8 and 10, they are describing us measuring the depth of the yellow agar after 10 and 20 minutes. So it's about measuring distance, really. So what the issue with this is it's simply just a matter of being difficult for it to, it's simply just a matter of it being really difficult to judge where exactly to measure the diffusion distance, because okay, in diagrams it might be a distinct line, but in real life I'm sure the lines are blurred between the blue agar and the yellow agar, and it can be quite seamless and not as clear and visible. It then says to use our results at 20 minutes to estimate the concentration of our hydrochloric acid in an unknown test tube. Well, if we look at our 20 minutes column, we got a value of 18. 18 lies between these two, 14 and 9. So it's going to be between 0.2 and 0.02. So I just went with 0.10, and it's only 1 mark, and it's an estimate, so it doesn't have to be that accurate. But anything within that interval is fine. Okay, then it says to suggest 2 improvements to the procedure that would give you a more accurate value for your estimated concentration. So this is a 2 marker. For the first mark, you have to have mentioned this, or else you can only gain 1 mark maximum. And that is, you would have stated concentrations both sides of the estimate. So for example, we estimated that the concentration was 0.10 moles per decimeters cubed. Have one test a concentration of 0.09 and then one for 0.11. Test their diffusion distances, and if it still lies in between, then we're on the dot basically at this point. But if we choose a bigger range, then we just kind of navigate back and forth until we get to the right concentration. Then we also have other options. So more concentrations with narrower intervals. So perhaps instead of going for 2 moles per decimeters and then 0.2 moles per decimeters cubed, that's quite a big gap. So if we have more, so we can narrow that gap, then we're likely to end up with more accuracy because we have smaller intervals to choose from. Then second option, and obviously a go-to, is repeat and calculate a mean. We've only calculated the diffusion distance once after 10 minutes, once after 20 minutes, but we haven't done any repeats. So we might get a more accurate value if we do so. Then lastly, we can just do each concentration separately instead of doing them all at once and checking their depths after 10 minutes. It's not going to give as accurate results, so we could do it separately. Certainly more time consuming and not possible during exam, but that is obviously a good answer still. Then we are told that for our 2 moles per decimeters cubed of HCl, so our standard H, calculate the rate of diffusion over 20 minutes. Rate is distance over time. Distance for at 20 minutes for the 2 moles per decimeters cubed was 18 millimeters. And then our time, they said 20 minutes, so we'll just do 20 minutes. We obviously end up with 0.9. To gain this mark, the 2 marks, 1, we need to have correct value and units. 2, we need to have our measurement divided by 20. It then suggests a hypothesis that states that the rate of diffusion decreases over time. Show with a tick whether your result supports or does not support this hypothesis. I said that the result does support the hypothesis because the diffusion distance in the first 10 minutes is greater than that in the second 10 minutes. And by this, you could just go back to the table and take a look. Well, obviously, overall, as as time goes on, the diffusion distance increases. Of course, distance can't decrease. So that's a given. However, if we take a look for our concentrations, in the first 10 minutes, for example, for H, we had a 10 millimeter diffusion distance. After 20, that went up to 18. But in reality, in the last 10 minutes alone, only increased by 8 millimeters, and 10 is greater than 8. And we see this trend throughout 8 and 6, 6 and 3, 4 and 2. So really what's happening is the diffusion distance is still increasing in the last 10 minutes, but at a slower rate. Why might this be? Because obviously the concentration gradient is getting smaller and smaller as more diffusion takes place. The particles are more evenly distributed, less concentrated in an area, a single area. So it makes sense for this to be the case. But yeah, you don't have to have had this answer to get this mark right. If your results do not support the hypothesis, let's say the diffusion distance in the first 10 minutes, for example, was less than or equal to the diffusion distance in the second 10 minutes, then you would say, oh, my results do not support, and that's completely fine. Because 20 minutes isn't that much time, so it might not have decreased in that time. Then we're told to carry out we're told that an investigation was carried out on the effect of temperature on cell membranes by measuring diffusion of pigment and beetroot cells. So, student immersed discs of beetroot into water at different temperatures for 10 minutes, then use a colorimeter to measure the percentage of transmission of light through each sample of water. So basically we have water, beetroot disc, and then pigment would go out by diffusing through the cell membrane, and then a colorimeter would measure the transmission of light. As the pigment diffuses out into the water is going to be less clear, it's going to be more pigmented, and less light will pass through. So the colorimeter is going to record a lower percentage transmission of light. We're going to see how temperature affects diffusion of pigments, or the rate of diffusion of pigment. So as you can see, our temperature is our independent variable. It's the one that's changing. And the dependent variable, the percentage transmission of light, is the one that's being affected, or the effect, or what's being produced. Okay? We're told to plot a graph based off of the data. And so we have. Now this is a 4-marker. The first mark is for labeling the axes and including the units degrees Celsius for temperature. The second mark is for the scales on the x and y axes. Both needed to be 20 degrees Celsius to 2 centimeters. So this is 2 centimeters, and labeled at least at each 2 centimeters. So labeled at each 2 centimeters at least. If you want, you can add the in-between ones. But it's not a must, okay? So same with the y-axis, exactly the same, but it'd just be percentage, of course. And really, when you're looking at scales, you should only be looking at numbers like this: 1s, 2s, 5s, 10s to go up in. Not too small of a scale, because you want your data to fit, but not too big of a scale that your graph is really tiny and doesn't cover at least half of the graph. Our third mark is for correctly plotting all 5 points, either using small dots or crosses. And our fourth mark is for joining them all with a thin line that passes through all the points. So I tried my best. Clearly there are some issues with my graph, but it should be as good as you can do, really. Then we are asked a question about temperature and its effect on the permeability of the cell membrane. So it says to state one reason for the change between 45 degrees Celsius and 60 degrees Celsius. So that is around here, when there's a very steep drop in the percentage transmission of light. So that would mean that diffusion is happening at a much higher rate. Well, if we're talking about permeability, really, talking about temperature, and we're talking about specific like range and a steep drop. What does this remind you of? Well, hopefully it reminds you of denaturation of proteins. Remember, the membrane is phospholipid bilayer, which contains a lot of proteins. Now what this means is temperature might denature the proteins, which can cause them to lose their shape, their structure, and thus it will leave gaps in the membrane, so there's more space for the pigment to kind of leave the membrane through. Secondly, you have the option of just saying that the phospholipids increase in fluidity. And then last but not least, cell membrane increase fluidity. Well, the cell membrane is made of phospholipid, so it's basically the same thing, but yeah.